Just tell me when you're ready. Okay. So welcome everyone for today's joint STG monthly meeting. We have Mikey as our presenter. And one thing about this moderator that we're having now, I'll just keep in the background and just let everybody do their thing. The reason we decided to have a moderator is in the end, when we have the discussion, to maybe help people get their voice heard during that ongoing discussion. So if there's people talking and you just want to ask a question or make a comment, you can use the raise hand feature. When you click on the participants list at the bottom, you see a little blue hand. You can click that. I will see that and I will interrupt whoever is discussing in a timely manner and help you to ask your question. If you can't find the raise hand feature, you can also type your question in the chat box. So I'll just keep an eye on raised hands and the chat box during the discussion. So with that, let's welcome Mikey and listen to his talk.
Hey, Gil. Thanks for coming, everybody. By the way, feel free to verbally interrupt me if you have questions as well. I won't object. I want to start by the same way that Ron Harris Warwick started last time, which is to apologize, because there's no way I could fit 30 years of work into 1 hour. So my task was to talk about projection neurons, and so that's what I'm going to do. Won't cover everything, but hopefully it will wet your whistle enough to be interested to go and read the associated papers. I started work on this project when I moved to Eve's lab back in 1984. And our first projection neuron paper came out in 1989. So I wanted to begin by either reminding you, or for many of you probably, informing you that before we started work on projection neurons in the crab, there were a few projection neurons that had already been identified and studied to some extent in a variety of different lobster species, including the well-known IVNs, which later on became the PS neurons in the European lobster, the anterior caloric modulator, or APM, and the commissural gastric neuron, CG. And as you can see here, they were studied in a host of different spiny lobsters, as well as in the bottom, the CG neuron was studied in Homarus gammarus, the European version of Homarus americanus. And most of these investigators are now either retired, passed away, or moved on to other systems, but one who hopefully is occupying one of these little squares on my computer monitor, and that is our very own Patsy Dickinson, who, along with Frédéric Nagy in the Moulin lab in Arcachon, France in the early 80s, identified and characterized the actions of APM, a projection neuron in the spiny lobster that lives in the esophageal ganglion. So as I said, when I started work in Eve's lab, there were no identified projection neurons yet that had been found in Cancer borealis, and that's primarily because Eve and her group had just started using cancer, 2 or 3 years before I got there, something like that. And so it was an adjunct to Panularis, which had been the work lobster, if you will, up until that point. And so when I was finishing my thesis work and had settled on coming to work with Eve in the STG, we decided that my focus would be to try and find the neurons that contain proctolin and that innervate the STG. And as you can see here in this snippet from my NRSA application in 1983, if you look at the red line here that I've underlined here down in the bottom, you can see that we were aiming to identify and characterize the neurons containing proctolin, which, as you probably know, was quite successful across the years. But the reason we picked proctolin as opposed to one of the other peptides to focus on is because Eve, Scott, and Kathy had already done all the basic background work with respect to proctolin. They had determined that there was proctolin-like immunolabeling with cell bodies stained in the commissural ganglia, in the, let me change this to a laser pointer, the commissural ganglia and the esophageal ganglion, and neuropil staining in the STG. And in addition, bath-applying proctolin had a nice, strong excitatory action, as you well know, on the pyloric rhythm. So when I came to the lab, we decided to start by doing backfills, dye backfills of the stomatogastric nerve in order to label the cell bodies whose axons were in the STN, thereby making them candidates for projection neurons that innervated the STG, and that we would wed those backfills with immunolabeling with antiproctolin antisera to see if we could find the proctolin containing projection neurons. And the dye we used, Lucifer Yellow, was one of the few, at this time, one of the few available fluorescent dyes that was small enough a molecular weight to diffuse effectively for some distance. And indeed, when we made a Vaseline well around the STN, filled it with a solution of Lucifer Yellow, and cut the stomatogastric nerve, the Lucifer Yellow diffused through the cut axons in both directions, staining a subset of cell bodies in the STG and staining cell bodies in the esophageal ganglion that projected into the STN. Unfortunately, the Lucifer Yellow didn't travel far enough to give us information about the commissural ganglion, but in the esophageal ganglion, as you can see here on the left, 2 small cell bodies and 1 very large cell body repeatedly labeled with the Lucifer Yellow backfills. The big one labeled CD1 is a motor neuron, cardiac dilator 1. It's very large cell body generates a very large axon, the largest one in the STN. It projects down to the STG, arborizes there, and exits out the ALNs to innervate cardiac sac muscles. That is proctolin immunonegative, but these 2 little guys here, which normally show up in the esophageal nerve instead of in the esophageal ganglion, here they are at higher mag, turned out to be proctolin immunoreactive and became the neurons that we know as the modulatory proctolin neurons, or MPNs. Panel C here is proctolin immunolabeling. Panel B is Lucifer Yellow labeling, as you can tell from this nice color image. For those of you who are curious about how the modulatory proctolin neuron got its initials to be the same as my initials, I refer you to Pierre Meyrand, who insisted that we name these neurons in this manner, as opposed to calling them the proctolin modulatory neurons, or PMNs. So we're going to come back and talk in some detail about the physiology of the MPNs, but sticking for a moment with the anatomy, once it was time for me to grow up and have my own lab, one of my first goals was to find a dye that would enable us to label the projection neurons in the commissural ganglion. And in between the Lucifer Yellow backfill work that we had done and starting my own lab, this small molecular weight dye biocytin became available. Now, biocytin itself is not autofluorescent, but if you fix and process the preparations after you do a backfill, you can tag it with a fluorophore and thereby image the stained neurons. And biocytin backfills became the first project of my first graduate student, Melissa Coleman. And using biocytin backfills, Melissa was indeed able to reproducibly stain 15 to 20 cell bodies in each commissural ganglion, as well as the MPNs and the OG, and some cell bodies in the STG. And we wanted to know, among the many things we wanted to know, is whether these 15 or 20 cell bodies in each commissural ganglion, plus the paired MPNs, plus the paired IVNs, represented just a few or many or all of the projection neurons that innervated the STG. And so to that end, we collaborated with Brenda Claiborne. And what Brenda did for us was to obtain a set of serial sections of the STN and in EM in order to enable us to count axon profiles in the STN and match the number of axon profiles on the STN with the number of neurons cell bodies that we labeled with our backfills. And to our delight, as it turned out, there was an extremely good match between the cell bodies labeled with our backfills and the axon profiles in the STN, such that it appears that most and possibly all of the projection neurons that innervate the Crab STG are local in the commissural ganglion, plus the MPNs and the IVNs. Now, Melissa, after doing this anatomy, which she will be the first to admit she hated doing at the time, but became the basis for everything my lab has done since, was able to go on, identify, and contribute importantly to the characterization of these 4 projection neurons, called the Melissa Coleman neurons 1, 5, and 7. And what started out as Melissa Coleman neuron 2, but as a result of a cranky reviewer, we changed its name to commissural projection neuron 2, or CPN2. And we'll talk in some detail about these 3 neurons as we go through, through the rest of the hour: MCN1, CPN2, and MPN. One of the additional things that Melissa did with her biocytin backfills was the same thing that I had done with the Lucifer backfill, and that is she double labeled these backfill preparations with various peptide antibodies. And doing so, she was able to determine that there are only 3 proctolin neurons that innervate the STG. And as our good fortune continued, thanks to the efforts of Dawn and Andy Christie, we were able to determine that MCN1, MPN, and MCN7 were those 3 proctolin-containing projection neurons innervating the STG. And Dawn then went on to spearhead the physiology characterization of these proctolin neurons and compare them to the effects of bath-applying proctolin. And this was an issue. To what extent does bath application mimic the effects of neural release of the same substance? Back in the early 90s was an issue that had really not been addressed in many or perhaps any systems. And so we set out to do this. I did the first component of this actually when I was still in Eve's lab, comparing the effects of bath applied proctolin with the effects of stimulating MPN on the STG. And as it turned out, the match was extremely good. The MPN was essentially acting like a proctolinergic neuron in terms of the way it excited the pyloric rhythm. Subsequently, this turned out to be a surprise because we found that MPN also contained GABA. And so despite the fact that it had this second transmitter, its effects on the pyloric rhythm were indistinguishable from those of bath-applied proctolin. Yet MPN does have some GABA-like actions, and they probably are mediated by GABA in the STG. And if anybody remembers at the end of the talk and wants to see a few examples of targets of MPN, which evokes PSPs on its target neurons in the STG, I'll be happy to show them. For now, though, the better focus is the work that Dawn did to show that the important GABA actions, at least those we know of MPN, took place instead in the commissural ganglion, where MPN uses GABA but not proctolin to inhibit the activity of MCN1 and CPN2, thereby preventing activation of the gastric mill rhythm. And it's not just that MPN uses GABA and not proctolin to suppress the activity of these 2 projection neurons, but both of these neurons actually are strongly excited by proctolin, just not proctolin from MPN. Because when Dawn used a GABA receptor antagonist to eliminate the GABAergic actions of MPN on these neurons, there was no response. So either MPN doesn't release proctolin in that neighborhood, perhaps not in the commissural ganglion at all, we don't know that, but with respect to how it affects these projection neurons, it's exclusively acting as a GABAergic neuron. MCN1 itself actually became a counterexample to MPN. So MPN acts equivalently to bath-applied proctolin, showing that neuronal release and bath application can, at least sometimes, be effectively equivalent. But MCN1 stimulation actually does not have the same effect of bath-applied proctolin because MCN1 stimulation also turns on the gastric mill rhythm, and it has another co-transmitter, CAB-TERP1A. And we'll come back to talk about that in quite some detail. So bath application does not always mimic neuronal release. And the third proctolin neuron is yet another example of the latter, not mimicking neural bath application, because MCN7 evokes a third type of motor pattern from the STG. Now, an additional bonus we got out of identifying these proctolin neurons was the fact that it gave us the ability to ask the question of whether different neurons innervating the same target circuit using the same co-transmitters affects that target circuit the same way or differently. And so to that extent, we were able to compare the effects of MPN and MCN1. And so for those of you who are scratching their internal minds about how we can do what I just said, given that MCN1 has CABTERP1A as an extra co-transmitter, it's because we were able to eliminate the actions of CABTERP1A with a tachykinin receptor antagonist, therefore turning MCN1 into a proctolin and GABA neuron. And by we, I mean my former postdocs, Debbie Wood and Wolfgang, who is again in a little square somewhere on this computer monitor. And what they were able to do then is to show that when they eliminated the actions of CABTERP, MCN1 no longer activated the gastric mill rhythm, and instead, like MPN, only excited the pyloric rhythm. However, the pyloric rhythm response to these two neurons was not the same. And Debbie and Wolfgang went on to show this using a number of parameters, but I'm only going to show you one of them here as an example. And so here on the left is simply our standard phase relationship for pyloric neurons during MPN, on the top here, during MPN stimulation, where, as you can see, across this normalized cycle period, as usual, PD and LPG are active first, followed by IC, LP, and PY, and MPN never activates VD in the isolated STG. MCN1 stimulation, if you just note all of the triangles here, those triangles all are locations where the phase relationships during MCN1 stimulation are distinct from those during MPN stimulation. And on top of that, as is evident, MCN1 consistently actually activates the VD neuron, while MPN does not. So despite using the same pair of co-transmitters, their effects on the pyloric rhythm are not the same, not only for phase relationships, but for the activity level of various pyloric neurons. So Debbie went on from this to address the question of whether this difference is due to some aspect of proctolin. That is, do these neurons release different amounts of proctolin, or perhaps they're differentially degraded by extracellular peptidases due to the location of their proctolin release relative to the location of the peptidases, etc. And to this end, she was able to take advantage of the presence of an aminopeptidase inhibitor, which Melissa had identified many years earlier, and study the effects of stimulating MPN and MCN1 at the same firing rate to eliminate that variable in the presence of this peptidase inhibitor so that there was no proctolin degradation. And when she did this, what she found was that these two, the pyloric rhythm response to these two neurons became more similar. What I'm showing you here on the left is just what I showed you in the previous slide as the sort of the control, MCN1 without CABTERP, spantide1 is the blocker, versus MPN stimulation. And so when Debbie did the same experiment, but with this peptidase inhibitor present to rescue proctolin from degradation, you can see by these little purple-like boxes that I've drawn in here, these pyloric rhythms became more similar. They didn't become identical, but they became more similar, not only in their phase relationships, but with respect to their firing frequency and burst durations and number of spikes per burst. So at least one aspect of the distinct way in which these two projection neurons affect the pyloric rhythm has something to do with differential actions of proctolin that they release. For the rest of the morning, the afternoon or evening for Abdel, we're going to focus primarily on MCN1. And MCN1 is a neuron that we've been able to record in 3 different locations. And so here you see the result of an intracellular difill of MCN1 cell body in the commissural ganglion. It has an elaborate arborization that's predominantly on the ION side of the commissural ganglion. And its axon projects through the ION and down to the STG, where as a result of being able to record MCN1 intraaxonally at the entrance to the STG and fill it with dye, you can see that it arborizes quite extensively within the STG neuropil. In the first set of MCN1 related stuff that we'll talk about, most of the experiments are done with the commissural ganglia cutaway in order to standardize the prep, eliminate the variability in the background activity of COG projection neurons. And we're able to do this because there are only 2 COG projection neurons that go to the STG via the ION, MCN1 and MCN5. And MCN1 has a larger diameter axon, lower threshold for extracellular stimulation, and we're able to thereby stimulate MCN1 in the ION selectively. And so in this first set, I simply want to lay the groundwork of how MCN1 affects the gastric mill rhythm. And so here we have the intracellular recordings of LG and interneuron 1, the reciprocally inhibitory pair that are at the center of gastric mill rhythm generation, as you'll see momentarily, and AB, because AB has this functionally important inhibitory synapse onto interneuron 1. And here's the control before MCN1 stimulation. As usual, AB is oscillating. INT1 is in pyloric rhythm time because of the synapse from AB, and because INT1's resting potential is above threshold for spiking, and so INT1 is always active except when it's being inhibited. And LG is a boring neuron unless it's provided with interesting properties, so LG is silent. When Melissa stimulated MCN1, here just stimulating it tonically, as you can see, the gastric mill rhythm, a nice stable gastric mill rhythm, is elicited, and AB's cycle frequency increases. There's a fast excitation of INT1 and the slow depolarization of LG, and then we have the standard rhythmic alternation between LG and interneuron 1, with LG representing the protraction phase and interneuron 1 representing the retraction phase. And so Melissa went on to determine how MCN1 affected each of the gastric mill circuit neurons. And the way this little circuit schematic is drawn, the protraction phase neurons are on top, the retraction phase neurons are on the bottom. And with respect to rhythm generation, the color code, as you see in the little box here, is that these 3 neurons, MCN1, LG, and interneuron 1, are necessary for rhythm generation. AB regulates the rhythm, and all the neurons in gray, you can hyperpolarize them and they don't change the ability to generate the rhythm, but they will alter the pattern, and they're all motor neurons, of course, that innervate gastric mill muscles. So they're functionally important, they're just not necessary to generate the basic rhythm. And as I hope you all appreciate, the connectome is necessary but not sufficient, and there's no way one could understand how this system works just by looking at this. And so, fortunately, we were able to whittle this down to our interest, which is to try and understand rhythm generation by focusing initially on just these 3 neurons, although just these 3 neurons have enough synapses between them to still make it daunting to understand by looking at the circuit diagram how this circuit diagram produces the recordings that we see here on the right. In collaboration, though, with our pal Pierre Meyrand, we were able to determine rhythm generation and the fact that the way in which this little circuit works has 2 completely different personalities depending on whether it's the retractor phase or the protractor phase. So here, during the retractor phase, which we'll just identify with this purple square at the beginning of the stimulation, when interneuron 1 is active, as I already mentioned, there's a fast increase in INT1 activity, and LG is slowly depolarizing. And so during this time, MCN1 is releasing transmitter. It has an ionotropic excitatory action on INT1, hence the fast activation of increase in activity of INT1, and a slow metabotropic excitation of LG underlying this gradual depolarization. When LG eventually depolarizes sufficiently to reach spike threshold and begins to fire, it immediately inhibits with a very strong synapse and turns off interneuron 1. And also, it eliminates further transmitter release from MCN1 due to the fact that it has an ionotropic inhibitory synapse onto the STG terminals of MCN1. And much to our surprise, when we originally did this, the electrical synapse between MCN1 and LG is basically non-functional during the retraction phase, but becomes effective and strong during the protraction phase. So even though LG is no longer being driven by metabotropic excitation, that metabotropic excitation that built up during retraction only decays slowly, and it's buoyed by this electrical EPSPs. So the LG burst persists for about 5 seconds until the metabotropic action has decayed sufficiently that the electrical EPSPs no longer reach spike threshold. And at that point, we return to the retractor phase. So the way in which that little busy circuit operates is with MCN1 acting to activate the circuit during retraction by a neurotransmitter release, and during the protractor phase by electrical coupling. Now, if we focus on the retraction phase when MCN1 is releasing its co-transmitters, it turns out that the ionotropic excitation of interneuron 1 is via GABA, while the metabotropic excitation of LG is via CABTER, with proctolin affecting neither of these neurons. So as it turns out, as was determined later on by Wolfgang, interneuron 1 is strongly excited by GABA, but is unresponsive to both proctolin and CABTERP. LG, which is excited by CABTERP, is unresponsive to proctolin and is responsive to GABA. It's inhibited by puffed GABA, but that must be due to GABA receptors that enable LG to respond to other GABAergic neurons, because there's no GABAergic synapse from MCN1 onto LG. Now, this metabotropic action of MCN1 onto LG is actually due to that inescapable current that we hear about every time we meet, and that's IMI. And what we're seeing here down below is part of a computational model that Nick DeLong produced based on an earlier model that we'll talk about in a few minutes from Farzan and Yair. And I'm using this here just to emphasize the fact that the way in which this cycling occurs is that during the retractor phase, when MCN1 is releasing its co-transmitters, there's this steady buildup of the IMI conductance, GMI, in LG, because MCN1 is continually releasing CABTER. As soon as LG begins to fire and feeds back to inhibit further transmitter release from MCN1, GMI decays. And this decay is such that the LG membrane potential remains above threshold for some period of time, which essentially defines the LG burst duration, due to the fact that the electrical EPSPs that we talked about are rising off this, although declining, still super threshold level of membrane potential, so that LG can maintain its firing. But when the GMI reaches a particular threshold point, the electrical EPSPs cannot reach spike threshold. The GMI conductance drops and the LG membrane potential drops, and we return to the retractor phase.
Mikey, can I ask you a question?
You just did.
Second question, does the electrical synapse play any additional role than extending the duration of the burst?
Any additional role other than extending the duration of the burst?
I assume that that's what you're saying it does, right?
It does. It does do that. And I can't say it doesn't play any additional roles, but we don't know. Either we don't know of any additional roles or I'm not remembering them. Anybody else remember an additional role? Okay, none of us remember an additional role, but it's certainly true that while the experiments that we've done all confirm our working model, it doesn't mean that we fully understand how this works, and it would not surprise me if there's more secrets to be learned. Come and do a sabbatical post-COVID. Okay, so as I mentioned when I was showing you the fill of MCN1 and the STG, we're able to make intra-axonal recordings of MCN1 at the entrance to the STG, which is why we're able to establish the fact that this feedback synapse is present, that it's ionotropic in nature, and that it regulates MCN1 transmitter release. And so these intra-axonal recordings are quite close to the entrance to the STG, and they enable us to, record unitary IPSPs in MCN1 just as if we were recording from a STG cell body. And I have to say, and it's actually quite, I just lost the word because I need some coffee, beneficial that Jorge just asked a question, because there he is. Because if it weren't for Jorge, we would never have been able to do all of the work that we've done with these axon recordings, because it was Jorge who did them first. Because there was a point in time during his graduate work when he was doing something even more difficult than making intra-axonal recordings, and that is he was trying to make neuropil recordings of the LP neuron to obtain biophysical data of LP for a model they were developing. And he was pretty frustrated one day, and I remember him saying that he was just fed up and he's just going to go stick axons, which he did. And he came over to my rig and said, "Hey, look, it worked." And so he was able to readily impale axons at the entrance to the STG, but it was not something that he wanted to pursue. And he graciously agreed that it was fine for me to continue doing it, which I did continue to do. So thanks, Jorge. Couldn't have done it without you. So here's a recording of an intra-axonal recording of MCN1, just to give you an example of what it looks like when we make these recordings. And so in this particular recording here, we're injecting a little bit of depolarizing current into MCN1 so that it'll fire. You'll note it's not firing tonically. It's actually being rhythmically interrupted in pyloric time due to feedback from the pacemaker neurons. And here in the middle, as you can see, we've injected some depolarizing current into LG to show that LG inhibits MCN1 effectively enough to eliminate its action potentials. And if you go to Melissa's paper listed down here at the bottom, you can see figures of the unitary IPSPs that underlie this inhibition. There's just not enough time to cover everything here. But what I wanted to do is emphasize the fact that this LG synapse onto MCN1 and the STG locally regulates MCN1. It's having no effect on MCN1 back in the commissural ganglion because it's just electrotonically too far away. And one example of that is shown here. So here we have a prep where the commissural ganglia remain connected. Here's an intracellular recording of the right MCN1 cell body, which we've depolarized to drive a gastric mill rhythm. You can see that MCN1 again is not firing tonically, but rhythmically in time with the gastric mill rhythm and the pyloric rhythm. And we'll be back to discuss this feedback from the STG that underlies this pattern later. The point I want to make with it now is the fact that if we look at simultaneously at an intra-axonal recording of the opposite MCN1, which is not active, we can reveal that LG is still providing this rhythmic inhibition in the STG that's not interfering with MCN1's firing in the commissural ganglion. So at times when MCN1 is inhibiting, at times when LG is inhibiting MCN1 transmitter release in the STG, MCN1 is still free to release transmitter in the commissural ganglion. All right, so we're going to come back to this story in a bit. First, we're going to tell Adobe Acrobat to go away. Go away, Adobe Acrobat, go away. Well, we can tell it, but it doesn't seem to want to. Oh, this is super annoying. I can make it go small though. Okay. What I want to do next is to shout out thanks to Farzan and his partner in crime, Yair Manor. when Farzan and Yair were postdocs in Yves' lab, they contacted me because they wanted to try and develop a model of the gastric mill circuit, of the gastric mill rhythm generator. And so they asked Melissa and I to send them all of our physiology data for the gastric mill circuit so they could see if they could put it together in a model, which we did. And to our delight, or at least they claim that it was super easy to do. That is, that without a lot of tinkering, their model readily generated a gastric mill rhythm. And using their circuit model was essentially all of the synapses that we had previously identified and that I just went through with you. And as you can see here on the top is an example of the biological gastric mill rhythm with MCN1 stimulation. And on the bottom is the model version of the gastric mill rhythm with MCN1 stimulation. And the great thing about this model, which we have carried forward through the decades to great success, is the fact that it's made a number of predictions that have provided considerable value to us and allowed us greater insight into how the system works. And while the fact that I don't need to preach to this choir about the value of models, what I want to emphasize is the fact that the predictions the model made were readily testable by us in the lab. And one of the first predictions of the model that we went on to examine is that the model told us that the AB neuron played a really important role in gastric mill rhythm generation. And now, you know, in retrospect, one might wonder why we couldn't figure that out for ourselves, because it's obvious in retrospect. But as I'm sure you all appreciate, there are so many possible experiments to do that having a reason to do a particular experiment instead of just floundering around and fishing until you find something that's worth pursuing has a lot of value. And so the model predicted that AB had a substantial impact on the gastric mill rhythm. And so Marlena Bartos, who you heard from at the STG meeting in her new life as a hippocampal big shot, was a postdoc in the lab. And in collaboration with Farzan and Yair, Marlena took the predictions of the model and simply hyperpolarized the PD neurons to shut off AB in the pyloric rhythm and ask what happens to the MCN1 driven gastric mill rhythm. And as you can see here, gastric mill rhythm slows down. So on the left, the pyloric rhythm is obviously on. On the right, same prep, but with the pyloric rhythm off. You see in the PDN, there's no activity. MCN1 has, the LG neuron bursting has slowed down considerably, and there's no pyloric rhythm subthreshold oscillations in between the bursts. So the model predicted that this was a consequence of the AB synapse onto interneuron 1, which is what I emphasized earlier, but we need to know that was actually true. And at the time, it wasn't clear, it wasn't explicitly clear that the AB synapse onto interneuron 1 was the AB synapse that was having this impact on the gastric mill rhythm. And that's because, as I know many of you know, AB is an octopus. It has lots of synapses all over the circuit. And given that this is a network with lots of interconnections, a priori, while the AB synapse to int1 is the direct one, any one of these others, or maybe many of these others, could have been playing a role in AB regulation of the gastric mill rhythm. And so we took advantage of the dynamic clamp. And by we, once again, I mean Marlena, in collaboration with Farzan and Yair. And what they did was, as you see here on the left, was again to turn off the pyloric rhythm. No dynamic clamp on the left side here. Dynamic clamp is off. And we have slow but regular alternation between LG and interneuron 1. The gain here is pretty low, but this thickening of the int1 line here, those are action potentials. As you can see, not only is the rhythm slow here, but the DG activity is a little bit beat up. It's not a coherent burst. It's bursting repeatedly in between each LG burst. And the MG neuron, MG is labeled here, has lost its pyloric rhythm timing. To test the hypothesis that it's the AB synapse onto int1 that's responsible for the regular gastric mill rhythm, Marlena injected an AB-like synapse into int1. And when she did that, everything was good again. And so here you could see in the dynamic clamp trace that she's injecting rhythmic hyperpolarizations into interneuron 1. And that, as you can see, has reproduced pyloric timed oscillations in neuron 1. And you'll note that there's now pyloric timed events in LG and MG, but there's no pyloric timed events being injected into these neurons. These are a secondary consequence of the activity in INT1. INT1 inhibits LG and therefore produces this pyloric rhythm pattern, and INT1 inhibits MG and produces a pyloric rhythm pattern in MG. And the DG burst is now coherent once again. The pyloric rhythm is fast, and everything looks like a normal pyloric rhythm. And it was manipulations such as this which enabled us to be able to conclude that the AB neuron synapse onto interneuron 1 is the AB synapse that's most relevant for gastric mill rhythm generation. Now, not to be outdone, not only does the pyloric rhythm regulate the gastric mill rhythm, but the gastric mill rhythm regulates the pyloric rhythm. And you can see that here, that is during each LG neuron burst, the pyloric rhythm slows down. And we know it's due actually to LG activity because at this point, Marlena delayed the next LG burst by injecting a little hyperpolarizing current. And during that time, at the expected interval, there was no slowing of the pyloric rhythm. But it slowed once again when the hyperpolarizing current was removed from LG. And this rhythmic inhibition of the pyloric rhythm by LG, or slowing, I should say, is due once again to the fact that LG inhibits the terminals of MCN1, inhibiting MCN1 transmitter release, so that when MCN1 is active, when LG is active, it's no longer, MCN1 is no longer able to excite the pyloric rhythm. When LG is silent and MCN1 is releasing its co-transmitters, MCN1 is driving the pyloric rhythm. So we have, it's working both ways. The AB neuron is regulating the speed and pattern of the gastric mill rhythm, and the gastric mill rhythm is regulating the speed and pattern of the pyloric rhythm, even though they're operating in very different time domains, being about 10 pyloric cycles per each gastric mill cycle. All right, so with that kind of abbreviated primer on MCN1 rhythm generation in the isolated STG, I'd like to move back to a more integrated system and have the commissural ganglia reattached to the STG, and now focus on not only MCN1, but its partner CPN2. And in particular, two pathways that co-activate these neurons, the so-called ventral cardiac neurons, or VCNs, and the post-esophageal commissure neurons. But before we get to those effects, let me just show you how CPN2 differs from MCN1 with respect to the gastric mill neurons. So I've already told you that MCN1 excites all these neurons, and here it is again. In contrast, CPN2 actually inhibits three of these neurons, AM, DG, and MG, and drives the LG neuron and GM by electrical synapses. And interneuron 1 has a functionally important, inhibitory action on CPN2 in the commissural ganglion. So one thing that was initially perplexing until we thought about it and it became evident is that despite MCN1 and CPN2 having opposing synaptic type actions on some of these neurons, it doesn't matter because they're effective at different times. During the retraction phase, when INT1 is active and LG is silent, MCN1 is releasing transmitter, as we've already talked about, and so excites all these neurons. But CPN2 is silent because INT1 not only inhibits LG, but it inhibits CPN2. So CPN2 is limited to the protraction phase. During the protraction phase, MCN1 is not releasing transmitter because LG is inhibiting it. And during that time, CPN2 is free to have its synaptic actions on these neurons. So with that as a quick backstory, let's look at these gastric mill rhythms where MCN1 and CPN2 are co-activated. The VCNs were initially identified and characterized by former grad student Mark Beenhacker Work was able to show that the VCNs are a population, bilateral population of about 60 stretch-sensitive mechanosensory neurons that are not muscle stretch-sensitive neurons, but actually are embedded in the stomach lining in the posterior aspect of the cardiac sac. And their axons project through the VCN nerve and the DPON to arborize in the commissural ganglion, where they cause a long-lasting excitation of MCN1 and CPN2. And as a result, a relatively brief stimulation of the VCNs, eliciting a long-lasting activity of these two neurons, drives a gastric mill rhythm that can sometimes run for an hour or more after a 1 or 2 minute stimulation. The POCs were initially identified by Farzan when he was doing a sabbatical in my lab. And he was hunting along the commissure to find nerves to stimulate that might have an effect on the gastric mill system. And the very first nerve he stimulated had the POCs in him, and so he stopped looking for other nerves. And like the VCNs, the POCs also co-activate MCN1 and CPN2 for a long-lasting duration, driving a long-lasting gastric mill rhythm. But the VCN triggered gastric mill rhythm and the POC triggered gastric mill rhythm are quite different. And let's look at them. And so here we'll look at them with intracellular recording of PD for the pyloric rhythm monitor, an extracellular recording of LG, an extracellular recording of MCN1, and an intracellular recording of CPN2. And here, if we look after a POC stimulation, we see a gastric mill rhythm in which MCN1 and CPN2 are activated and fire regularly in pyloric rhythm time. And this is due to feedback from AB and interneuron 1, primarily AB in this case. And as you see, the resulting gastric mill rhythm, which we're characterizing here just by the pattern in the LG neuron, is such that each LG burst is subdivided into pyloric timed burstlets that are co-active with MCN1 and CPN2. And that's because MCN1 and CPN2 are driving this LG activity. And so their pyloric rhythm timing is LG's pyloric rhythm timing. And in between the LG bursts, interneuron 1 is active. You get a very different motor pattern when the VCNs are stimulated. As you can see here on the right, after VCN stimulation, the LG neuron burst is strong and tonic, not pyloric rhythm timed. The MCN1 burst is strong and tonic during protraction and is pyloric rhythm timed during retraction. And the CPN2 burst is high frequency firing and tonic during the protraction phase and basically off during the retraction phase. So different motor patterns that result from co-activation of the same two projection neurons. And I neglected to say that it was Mark who established that the VCNs co-activated MCN1 and CPN2, and they were necessary and sufficient for this motor pattern, while it was Dawn who went on to show that it was MCN1 and CPN2 that contributed to the POC version of the gastric mill rhythm. So how do they do this? Co-activating the same two neurons, but evoking different activity patterns from them. Well, as it turns out, both the VCNs and the POCs cause a direct, long-lasting excitation of these two projection neurons. Where they differ is the way they affect the AB synapses in the commissural ganglion onto these projection neurons. The VCNs, through an indirect means, by strongly activating some commissural ganglion neuron, yet to be determined, but secretly believed to be CPN2, is that it strengthens inhibition of AB during the protraction phase, which is what eliminates the pyloric timing, because the AB synapse is inhibited by this pathway, preventing AB from inhibiting these neurons. In contrast, the POCs appear to directly strengthen the AB synapse, enabling MCN1 and CPN2 each retain their pyloric rhythm timing continuously. And these two different activity patterns and these two projection neurons are then imposed on the LG neuron and result in these different gastric mill activity patterns in the STG, which in fact are then subsequently imposed on the muscles in the teeth. And this, of course, has always been an issue for us and other systems where the work is studied primarily in the isolated nervous system. Are we looking at different patterns in the ganglion that actually come together to produce what turns out to be the same pattern in vivo? Because obviously in the isolated system, we're missing a lot of components. We're missing a lot of sensory feedback. We're missing hormonal modulation. We're missing other pathways. And so fortunately, we were able to take the system, this system with the VCNs and the POCs from the dish to the animal and determine whether or not these rhythms were different or not in vivo. And this was a happy consequence of the fact that Wolfgang got a job in the United States. And what I mean by that is that at the time Wolfgang was preparing to move his lab from Germany to the US, he had a senior level graduate student, Florian, who didn't want to lose 6 months at the end of his thesis by just tearing down the lab and setting up a lab. And consequently, Wolfgang asked if Florian could come to my lab to continue his thesis work while Wolfgang moved the lab to Illinois. And so that's what Florian did. And in collaboration with my graduate student, Rachel White, we put together a nice story where Rachel did an extended analysis of the VCN and POC gastric mill rhythms effect on LG innervated muscles. Did the LG innervated muscles respond differently with different patterns during the VCN and POC rhythms? And Florian, as I'll show you in a moment, did a beautiful series of endoscope videos of teeth movements after eliciting VCN or POC rhythms in the crab. There's no time, I'm sorry, Rachel, for your muscle work. Everybody listening should go read the paper, the Deal et al. Paper. But what I will show you is an image from Florian's endoscope study. And in this work, what he did was to maintain an extracellular recording of the LVN, as you see on the top here, so that he could monitor LG activity indicated by the black bar, while taking videos of the lateral teeth. And as indicated by these labels, this gray dashed line here is the midline. And during the LG bursts, the two lateral teeth come together at the midline. That's the protraction phase. And when LG is silent, the teeth move apart again, back towards the interior lateral walls of the gastric mill. That's retraction. As you can see here by these arrowheads, during the LG burst, the two teeth come together relatively smoothly, not much evidence for pyloric rhythm timing. In fact, these black dotted lines here represent the PD bursts to give you some indication of the pyloric rhythm inside the gastric mill rhythm. When he stimulated the POCs instead to elicit a POC gastric mill rhythm, now you can see that LG bursts are pyloric rhythm timed. They're co-active with LP basically. And now if we look at the movements of the lateral teeth, they have pronounced pyloric timed sub-oscillations of the teeth. So it's more like a chattering movement instead of a smooth protraction. So in vivo, the VCN and POC rhythm, gastric mill rhythms, still generate not only different neuronal activity patterns, but different tooth movements. Why the crab needs these different tooth movements, you're welcome to ask, but you know that I'll be able to tell you is I'd like to know, but I have no idea. But if somebody has some idea, like for instance, maybe they're used for talking, we can find that out later. All right. I don't know what time it is. How are we doing here? Oh, we're doing fine. I must be talking fast. So in this last section that I want to cover, I thought I'd talk for an extended time over one topic and give you a sense of how much flexibility there can be in even a single sensory pathway's ability to affect the system. And for that end, we're going to talk about the gastropyloric receptors, or GPRs, a pair of muscle stretch sensitive neurons that were initially studied with physiology by Paul Katz and Ron Harris-Warwick's lab, but which Ron didn't have time to talk about when he was presenting last month. I'm sorry, Paul, we're not going to talk about your work either, but I wanted to give you a shout out. Paul's work was focused on the GPR effects on the STG, but we're going to go up to the commissural ganglion, where Dawn was able to show that brief GPR stims elicit a lasting excitation of MCN1 and CPN2 once again. As you can see, the GPR stimulation causes a relatively short-lasting depolarization and greatly prolonged activation of MCN1, while GPR stimulation causes a more longer delayed but eventual strong burst in CPN2 in the commissural ganglion. Now, interestingly, in the STG, when we look at an intra-axonal recording of MCN1, we see that GPR inhibits MCN1 in the STG while it excites MCN1 in the commissural ganglion. And while this might seem counterproductive, note that the duration of the GPR inhibitory effect is much briefer than the duration of the GPR excitatory effect. And now we've seen the way these individual actions on these two projection neurons operate, or not operate, but occur. Let's see what GPR does in the context of gastric mill rhythms. So here, as Dawn originally showed, rhythmic stimulations of GPR, which sort of mimic the timing that when it might be active during rhythmic movements of the teeth. Remember that GPR can be activated by DG neuron bursts, because when DG is active, it causes a contraction of the GM4 muscle, which shares an ossicle with the muscles innervated by GPR. So when DG is active, it stretches the muscle that GPR lives in, activating GPR. So GPR is active during the retractor phase. And so we stimulated it in a retractor-like pattern. And when this occurred, as you can see here, there was a repeated activation of MCN1 and CPN2. I'm not showing you CPN2 in this recording, but it's reflected in this activity of GM, which is a consequence of CPN2 activity. And so this rhythmic GPR stimulation elicits a nice gastric mill looking event. What does GPR do though during a gastric mill rhythm? Since, as I just said, GPR should be active during an ongoing gastric mill rhythm. So to address that, Mark depolarized MCN1 and the commissural ganglion. So again, here now we're working with commissural ganglion connected preps. Here's an intracellular recording of MCN1 cell body. Mark depolarized MCN1 to drive this gastric mill rhythm. You can see LG and DG are alternating. And he gave this 5-second or so duration GPR stim. And as you can see, the consequence of this stimulation was that the duration of the retractor phase when MCN1 is tonic and when LG is active was greatly prolonged. Actually, not just for the cycle when GPR was stimulated, but for several cycles thereafter. Oh, maybe I labeled this. Look, I labeled it. So here we can see that the control LG and MCN1 duration, if we map that same color duration here, you can see that the bursts are prolonged. And if we do the same thing for the retractor phase, there's a slight prolongation of the retractor phase as well. So during MCN1, CPN2 gastric mill rhythms that result from MCN1 direct activation, GPR's action is primarily to slow the gastric mill rhythm by prolonging protraction. And the reason I'm emphasizing that it's prolonging protraction is that when we look at an MCN1, CPN2 gastric mill rhythm that's triggered by VCN stimulation, we see the exact opposite effect. Instead, GPR selectively prolongs the retraction phase, having no effect on the protraction duration. So here, as Mark showed originally, we have an ongoing VCN triggered gastric mill rhythm. LG and GM are coactive and alternating with the DG neuron. And with these black lines, Mark stimulated GPR. And as you can see, the DG neuron bursts are greatly prolonged, the LG neuron bursts not so much. So there's a selective increase in the duration of the retractor phase, and there's no, excuse me, there's no extended effect. Once the GPR stimulation is terminated, the rhythm goes back to the pre-stimulation levels. So short duration effects that selectively affect the retractor duration. Interestingly, this turned out to be a consequence of an occlusion effect of the VCNs in the commissural ganglion. So during the VCN gastric mill rhythm, GPR stimulation had no effect on MCN1 activity. And what you see here down below, if you can see this legend here on the right, which might be obscured by the names of boxes of the audience, the white and black boxes are the pre and post GPR controls, and the gray box is during GPR stimulation for the firing frequency of MCN1 during protraction and retraction. And as you can see, MCN1 firing rate did not change, while the gastric mill cycle period was prolonged because of this increased retraction phase duration. Ditto for CPN2. During the protraction phase, when CPN2 is active, its firing rate was not changed, even though the gastric mill cycle period was prolonged. There's no retraction analysis for CPN2 because it's not active during VCN retraction. So, unlike the MCN1 CPN2 gastric mill rhythm that occurs without VCN interference, where the protraction duration is prolonged, during VCN stim, the retraction phase is prolonged, and the reason for the selective prolonging of the retraction phase is because the GPR actions in the commissural ganglia are occluded by the previous stimulation of the VCNs, and the GPR action on the gastric mill circuit results entirely from its inhibitory synapse on the MCN1 terminals in the STG. So we have a state-dependent effect of GPR. It can activate the gastric mill rhythm. It can modify the protraction phase of gastric mill rhythms in which the VCNs were not recently active, or if the VCNs are recently active, it selectively affects the retraction phase by only its synapse in the STG. Now, it turned out that we weren't done because it didn't make any sense to us that the effect of only this synapse in the STG would be to selectively prolong the retraction phase. And that's because it's inhibiting MCN1's transmitter release, and so it should be equally affecting, or if not equally, at least co-affecting MCN1's actions on both interneuron 1 and LG. And so, for starters, Nick DeLong, in a computational model, asked the model what would happen if we did just that, stimulated GPR during this gastric mill rhythm, where the synapse was placed on the primary axon of MCN1 entering the STG, so it reduced MCN1 transmitter release on both interneuron 1 and LG. When he ran that model, what he found was a result different from what occurred in the biological prep. The retractor phase was not prolonged selectively. As you can see here, the protraction phase was abbreviated as well. In fact, more abbreviated than the retractor phase. And Nick went on to do a set of physiology experiments to address the same issue by injecting a little hyperpolarizing current in interneuron 1 while stimulating GPR. And when he did that, I'm not going to show you the data, but you could look at his paper, he got the same result as he got here in the model. But if instead of this, he placed the model synapse so that it only weakened MCN1 excitation of LG and didn't affect MCN1 excitation of interneuron 1, the model outcome was the same as we saw in the original physiology of GPR stimulation during the VCN rhythm. And when he went on to do a set of physiology manipulations, he was able to establish that, in fact, this is the appropriate placement of the synapse. That is, that GPR selectively inhibits neuropeptide release from MCN1 without affecting GABA release from MCN1, with the consequence that the GPR stimulation selectively prolongs the retractor phase because INT1 is still being effectively excited by MCN1, But because there's less CAB-TERP release per unit time, it takes longer for IMI to build up and allow LG to escape from that INT1 inhibition and fire its burst. And just as a side note, because it's obviously right here on the image and it's in the same paper, while GPR has 3 co-transmitters, serotonin, acetylcholine, and elatostatin, by using a serotonin receptor antagonist, Nick was able to show that the GPR action on MCN1's axon terminals was entirely due to serotonin. Another example of divergent co-transmission. So with that, I'm going to wrap up and give you a couple of concluding points, some things for somebody to do in the future, and then see if anybody has anything they want to say or ask. So as I showed you already, we've been able, way back in the last century, to identify a subset of the projection neurons that innervate the STG and that influence the pyloric and gastric mill rhythms, most of our focus being on the gastric mill rhythm. And in particular, we focused on a set of inputs that influence the projection neurons and enable them to influence the STG circuits, including the ventral cardiac neurons, the post-esophageal commissure neurons, and the GPRs. And what I aimed to show you this afternoon was that we addressed the question, which, while relevant back in the early 90s, remains equally relevant in most systems today, the issue of whether BAF-applied modulator, whether it's a peptide or an amine or a muscarinic agonist, whether BAF application of a modulatory action effectively mimics neuronal release of that same modulator. And as we showed you, the answer is it depends. In some cases, it can be yes. In other cases, it's clearly no. Although, just to emphasize, even when the answer is no, focal application or BAF application studies provide a lot of information that's necessary to eventually understand how the neuron will affect the circuit, because it enables you to be able to study in isolation the way each one of the co-transmitters would ultimately affect the system. Showed you that there's divergent co-transmission. MPN uses GABA to inhibit MCN1 and CPN2 in the commissural ganglion while it excites the pyloric rhythm in the STG with proctolin. MCN1 uses only CAB-TERP to slowly excite LG while it uses only GABA to quickly excite interneuron 1. There's some pretty complicated rhythm generation dynamics, even for a tiny little circuit with 3 neurons. And we went through that in some detail, how MCN1 is able to drive a gastric mill rhythm. And that part of this dynamic involves events that you would never be able to understand or learn about if you made only intracellular recordings from cell bodies of projection neurons, because there are functionally, pivotally important events that occur at the axon terminals of these projection neurons. At the end there, we were talking about the fact that even the same set of projection neurons can elicit different motor patterns from the same circuit, because modulation can enable those neurons to themselves generate different activity patterns under different influences. And lastly, as I showed you, single sensory pathways, such as the GPRs, can have state-dependent actions such that even though they're affecting the same set of neurons under different modulatory states, they can have profoundly different effects on the circuit. And with that, I'll just list a couple of some nexts for one or many of you to think about and maybe pursue in the future. The one that's bugged us for years is, why the heck does every pathway we stimulate elicit a gastric mill rhythm driven by MCN1 and CPN2 when there's about 15 other projection neurons in the commissural ganglion that clearly have some effects in the STG? Is it because there are other versions of the gastric mill rhythm? Is it because, as we learned recently, the STG circuits can be responsible for other behaviors besides chewing? Is it because chewing is an even richer behavior than we've seen so far? We actually know from snacks recordings that are not published but live in my office or on the computer that recordings from the axons of some other projection neurons have some pretty interesting effects on the STG. We just hadn't been able to record from them routinely enough to put together stories. So what other surprises will these SNACS recordings reveal? A bother that we've always had with this work is the issue which we tend to collectively ignore, which is how isolated is an isolated STG. If you eliminate, even if you cut the STN and you don't have the esophageal ganglion or the COGs connected to the STG, you have all those projection neuron terminals, and they are not dead. And they're not only not dead, but there's an enormous amount of dicoupling between the terminals of projection neurons and STG neurons, leading to the possibility that when STG neurons are oscillating, even in an isolated system, they could possibly be evoking transmitter release from some projection neuron terminals. Don't know that they do, but don't know that they don't. Oh yeah, and something that we've wanted to do for years and Wolfgang has made stabs at, but so far unsuccessfully, is it possible for us to somehow co-image the activity of the neurons in the commissural ganglion so that we can really be able to start recording, at least by imaging, calcium imaging or some other means, voltage sensitive dyes, the activity pattern of many projection neurons that go to the STG that presumably all receive, or many of which should receive feedback from INT1 and AB and therefore exhibit recognizable gastric mill or pyloric time patterns. So with that, let's wrap up. I've had a bunch of people in my lab, not a ton, but here they are. Once again, I apologize for all the stories I haven't told. But I really valued everybody's contribution through the years. It's been a great run. And Logan is currently in the lab, so the run's not done. And mostly, I would like to thank Cancer Borealis, who's been an awesome colleague through the past 30 years. And obviously, we couldn't have done it without it. As many of you know, every crustacean species that we have collectively worked with has different advantages and disadvantages. And one of the great advantages that Cancer Borealis provides that the lobsters do not provide just because of their structure of the anatomy of the system is our ability to do intra-axonal recordings in the STN, which is just, at least as far as we tried, impossible because of the fact that in the lobsters, the STN entering the STG, the nerve bundle is separated from the sheath, the nerve sheath around it. And consequently, it's just not possible to pin down the nerve in a way where we can get the axons to lie flat. But in the crab, as you saw in that image that I showed briefly, the axons lie flat like the fingers of a hand, and you can spread them out so that a subset of them are large enough to impale. And I should say by large enough, I mean the largest ones, such as MCN1 and CPN2 fortuitously, are 10 to 12 microns. So that enables us to find them and record from them pretty routinely. Lastly, I think the last word should go to Dawn, because Dawn has had the mixed fortune of working in my lab under so many different guises for so many years, and we continue to collaborate. And back in 2009, she found this Dilbert cartoon, which she stuck on my door, which I think effectively represents the relationship that all students have at some point in their career with their PI. And with that, I will close the show and open it up to all of you if you have anything you'd like to ask or say.
Well, thanks, Mikey. And yeah, so this presentation or this talk is open for discussion. Just go ahead and discuss and ask if you have a question. Oh, I already see Michael Gray has a raised hand, so go ahead, Michael.
Oh, wow. Great talk. I'm trying to figure something out in my head, and I'm putting two things.
Maybe that's the wrong place to figure it out.
Yeah, is that if I understand the model correctly, the depolarization that occurs in LG's downstate is induced by GPR stimulating MCN to reduce norproctilin. And when we see that downward slope on LG, that's a fading of IMI. Is that correct?
Hang on, because what you said made sense to me, but probably to nobody else. So let me go back to that figure. This is what you want, right?
Yeah, yeah. So that decay is controlled by the decay of IMI. But there's
Yes.
and that happens over about 1 caloric cycle. But there's an alternate picture.
Wait. The decay occurs across about 5 pyloric cycles.
I'm sorry, 1 gastric cycle, about 8 seconds, right?
Yeah, around 5 seconds actually, but yes.
5 seconds. And then you showed us that LG also modulates the caloric. It slows it down, but that seems to be less than 1 caloric cycle. Now, I know I'm way oversimplifying here, but does this decay time, how do we account for the difference? What I'm saying is that the decay time when it stops is a lot faster when it's modulating PD versus when it's decaying in LG. I mean, there's 1,000 things that could be, but in the simplest model in my head is that when the rhythm stops, it almost resumes instantaneously. Do you know what I mean?
No, because I was too busy changing slides, so I wasn't listening completely.
In this one, it seems to me the time for it to get back to its normal caloric rhythm is almost instantaneous.
Yes, it is.
So what I'm trying to do is resolve the difference. I mean, it sets boundaries for how fast the current can deactivate. But do we have any idea why there's a difference in timescales? Like one is less than a caloric cycle and one is
Yes. Yeah, hang on a sec. Let me go back to the other. Okay, so the question is, why does the pyloric rhythm become fast again within 1 pyloric cycle here at the end of the LG burst? And so you can't quite see it. Well, you can see it here on the right at the very end here. Both in the model and in our experiments, it's pretty clear that Gmi does not return to 0. It becomes very small, but it's not 0. And remember that the pyloric rhythm is not off, right? It's ongoing. AB is oscillating. And so my expectation is that AB doesn't need a lot of IMI in order for it to suddenly jump back up again. But we've never done that analysis. And so the real detailed explanation to your question is something that would have to be done. But you're absolutely right that the slowing of the pyloric rhythm, sorry, I have to jump back and forth. The slowing of the pyloric rhythm here is very fast, and that's presumably because it must be that the time constant of IMI's effect on AB is actually quite short.
Yeah.
And remember that, and so, we don't know that, well, actually we do know if we looked at the data, but I'll just say for this, that we don't know because we haven't looked at the data, what the time constant is for IMI in LG versus AB or PD.
Yeah.
Right?
Well, I was assuming implicit in my question is that I'm assuming it's the same for all cells, but it may be different. And that's
it doesn't have to be. Yeah, it doesn't have to be. It could be, but I don't know.
Yeah.
Jorge might know from his old work if he has a memory for different pyloric neurons, but I don't remember.
Cool.
Okay, moving on. Look, we're done. Nobody has anything else they want to ask?
Mikey, I have a question.
Hi, Abdel, go ahead.
Hi. You said that the electrical synapse is turned on during protraction. is that just because the synapse is rectifying, or is it an active mechanism?
That is a very good question and a little bit complicated, but I can tell you things. Let me just go to some recording that would be useful. I guess I don't really have the right recording here. Our descriptor for the electrical synapse here is that it's a bit unusual because it's a voltage sensitive, non-rectifying synapse. And what I mean by that is that the LG and MCN1 membrane potentials need to be oriented so that LG is more depolarized than MCN1 in order for the connection, the electrical synapse, to be strong. And it has to be, and the voltage difference has to has a filter so that action potentials are too fast to affect it. So that when LG is more depolarized than MCN1, the electrical synapse is strong. And when LG is more hyperpolarized than MCN1, the electrical synapse is weak.
Yeah.
And so when during the retraction phase, when LG is hyperpolarized, the electrical synapse is very, very small.
Yeah. And then another question is there any like feedback or what we call efference copy from the STG back to these projection neurons, back to the ganglia there?
Oh yeah, while you were thinking about that question, I was giving the second half of my talk. So here, for example, in this, as you can see in this schematic, interneuron 1 projects back to the commissural ganglion to inhibit CPN2, and the AB neuron from the pyloric system feeds back to the commissural ganglion to inhibit MCN1 and CPN2. And so that's why these projection neurons, which is also true in the vertebrate locomotor system, for instance, these projection neurons, once they activate the pattern, their activity goes from tonic to motor pattern rhythmic because of the feedback they're getting from the circuit. That was your question, yeah?
Yes, yes,
Okay.
that was my question.
Okay, that's my answer.
Yeah, thanks.
So Dirk briefly had his hand up, but not anymore. So I don't know, do you still want to ask a question or not?
Well, maybe some general comment question to Mikey, which is part of your questions at the end, sort of like the future lies ahead list that you had there, and some of the stuff that you said during the talk was sort of hinting at that it would be really nice to understand how the system actually works, and which of the projection neurons are on together, if the pyloric modulation of the teeth movements really matter, and all of these kinds of things. To which degree do you actually, or what is your intuition, to which degree that is important for the main selling points that the STG almost always had, which wasn't to explain how decapod crustaceans digest food, but which was learning about principles of circuit dynamics, neuromodulation, and things like that? Is that too bird's eye view?
No, but there is more than one question in there. So I was just trying to figure out which one you wanted me to answer, other than all of them.
I'd say all of them.
But that would require me to remember all of them. Let's start at the end because I remember the end. In terms of the value of what we've learned for other systems, for example, the work that we have been able to do with these intra-axonal recordings has revealed, a rich role for local synapses in enabling circuits to operate the way they do. And there's very little comparable work so far that's been done in other circuit systems, particularly in the vertebrate systems, other than the fact that It's known both from anatomy and physiology that there are many examples of axo-axonic synapses in the vertebrate CNS, but what their functional role is in enabling circuits to operate, particularly for projection neurons, that's a black box. And so a lot of the principles that are developed regarding how projection neurons enable circuits to generate patterns are based on activity in projection neuron cell bodies, with the assumption that that's all you need to do. You stimulate the neurons and they tell the circuit to wake up and do stuff. Whereas clearly what we learn here, which I would be shocked if it's not equally true in other systems, is that many projection neurons are not just activators of circuits, but they're circuit elements. You know, their axon terminals act like circuit elements because they receive feedback from the circuits locally, and that feedback, which changes the local pattern of those projection neurons, is necessary. It's pivotal to enabling circuit operation. So, you know, I think that things like that, the study of that we've been able to do with co-transmission in a circuit context, these are all things that either have, are, or will resonate in other systems.
That's not really what I meant, but it is important. Is it important that that's a realistic type of activation, that you really have a good understanding of how the whole system operates in nature, basically? Or is it enough to ask questions like, what if I isolate a stimulation of MCN1, or if I pair it up, no matter if that actually happens or not?
Got it. So in other words, why should the collective we have been working on reduced preparations for the last 50 years? That's really what you're asking. In terms of what's its value for understanding
Actually, I'm asking the other way around. My bias is the other way around, which is this isn't the most interesting behavior that you can study, but the advantage are the exact other ones, which is the general principles about circuit operation and circuit dynamics. But a couple of your questions were sort of in my head a little bit of STGology. It's like there's 15 more, and that's all kinds of things to look at to really understand, you know, do the teeth movements during the retraction phase or whatever, do they matter functionally, and all of that, which I always find not that interesting. But on the other hand, you know, the more you dig deep into a system, the more maybe interesting generalizable questions will emerge from that.
Sure. So clearly there are two kinds of approaches that you can take when you're studying a system. And I think that all of us, tend to use both approaches. One is the so-called STGology focus, which to some extent one needs to do in order to be able to study the system as a model system. And often the outcome of determining or characterizing what seem to be details of how the system operates is to enable you to derive from it more general principles that you didn't really have in mind when you started digging into those details. The other approach, of course, the one you mentioned, is to take a system and use it to address how an intact system works in a particular way. And, you know, so I've always thought that there's comparable value in both so-called bottom-up and top-down approaches. And to some extent, we all go to the approach that lends itself best to what we want to do or feel capable of doing, with keeping in mind the value of what we're doing towards general principles. But yeah, part of the reason I've included kind of STG detail-y things in the things to do next is simply because in order to understand how the pieces fit together, you have to know who the pieces are and what they do.
And then one last comment. If you're shit-talking the experimental accessibilities of other crustacean species, please distinguish between spiny and clawed lobsters.
Was I doing that? Was I shit-talking?
Yes. So,
yes.
So Mikey.
So sorry. Marie had her hand raised for quite a while.
I'm on mute now. This was just basically in response to Dirk's question, because I think Dirk wasn't there at the last meeting. I think if it holds up that the STG is also really involved in communication or sound production, then I think it's really, really valuable that we know all these details about when do the teeth touch, how much does the pattern change with different modulations, and is there a sense in the redundancy of different rhythms. So I think that it is extremely valuable and could become even more important as we understand more about the different functions of the system. So I don't think that the idea of saying, oh, we're just interested in the network and how the network works on its own without looking at the behavior is realistic in any system at all, and definitely not in the STG.
So I was sort of struck, Mikey, about your number one, why does every pathway elicit a gastric mill rhythm driven by MCN1 and CPN2?
Yep.
And it speaks to really interesting ideas about convergence of multiple sensory inputs onto some common drives. And on the one hand, you can imagine you have lots of different forms of gastric rhythms, but on the other hand, if you really have all this massive convergence, that decreases that effectively. So speaking about organizational principles, it's really very peculiar.
It is. In fact, that's why I have number 6 on this list, something that we just hadn't gotten to, which is, given they're all converging, it really behooves us to determine what the outcome is of co-activating in some appropriate pattern all these different pathways.
And that would actually argue it might be useful to know under which conditions those different sensory neurons are activated.
Agreed. Yes.
Together or not together.
Right.
So Dirk, that's an example of something where there might be some interesting circuit concepts that you wouldn't see if you didn't do all the STGology.
Well, I mean, there is this interesting possibility that there may be a completely different way to activate the system that isn't dependent on sort of classic sensory feedback from the stomach movements or just some chemosensory input or something, but I don't know, brain state or whatever it may be. So that some of these descending systems bypass that whole system. So all the sensory feedback that's relatively local that you have looked at will activate basically MCN1 and CPN2, but some of the other descending neurons may not be targets of any of that type of sensory feedback, at least.
And
Maybe
also,
it's something like voluntary teeth movements.
but also all the hormonal modulation, which has basically been washed away. And so it's possible that if you had a normal hormonal state, that some of these answers would look different.
Yeah, although so far when we have used hemolymph on the STG, we haven't really seen anything that lends itself to believing that other projection neurons are being activated, at least in the 2 conditions that we've looked at.
No, I don't actually mean that. I think it's that some of the answers you get might be different for different hormonal conditions.
Yes, that's true.
I think another thing is the temporal nature of the stimuli, because during stimulation of some of these pathways, we get activation of other projection neurons like MCN5, and we don't understand what the heck it's doing. And then it develops into a gastric mill rhythm that persists for a while. So I think there's the possibility that some of the other projection neurons might be involved in behaviors that have, that are needed for shorter periods of time or something like that. And without knowing what other behaviors there could be, such as communication or other things, then we can't answer that, because we see, things that don't look like rhythms to us that we recognize. So I think that's still another possibility for some of the other projection neurons.
Yeah, we certainly are limited by our lack of knowledge about the correct firing rates and patterns of some of these sensory pathways. And they do, and some of them at least do have different effects when you use different rates and patterns of stimulation.
Any further questions or comments? Now is your last chance. If not, Mikey, thank you very much for this awesome talk. Thanks to our organizers, Nelly and Smita. And I think the next meeting is scheduled for, I don't know, in two weeks or 3 weeks or something. Nelly, you're muted, but 2 weeks, okay.
In 2 weeks.
Yeah. Then we will hear from Ron Calabrese, if I remember correctly.
Yes, on December 14th.